Dear students, welcome to this MOOC. MOOC stands for Massive Open Online Course. In this MOOC, we shall be providing you a variety of multimedia enriched e-content, activities and assessments in a very learner friendly manner so that you are able to understand the various topics of this course very well. Let me now tell you briefly about the structure and main features of this MOOC. The entire MOOC has been divided into various weeks so that you can plan your learning in a very logical and systematic manner. It is advised that you must actively participate in the various components of this MOOC, that too regularly on a weekly basis. For every weekly schedule, you are first advised to watch 2 to 4 videos assigned to you per week. Then you must read the learning material provided as e-text and also go through other content in the Learn More section. After this, you are advised to carry out the various activities like taking part in the discussion forums, visiting the related web links which we have provided for you, attempting crosswords, etc., earmarked for each week. After this, to test your knowledge of what you have learned during this week, you must attempt the quiz in the assessment section for that particular week. I wish you all the best in this very interesting self-learning experience. Please remember, we shall always be there with you to provide you support for any problems you face. The MOOC coordinator along with junior faculty as tutor shall always be available to solve your queries which you can post in the discussion forums or you can send them to us through mail which shall be notified to you soon. We would be very happy to know your experience after the completion of this MOOC in the form of your feedback which will help us enhance the quality of this MOOC further. Dear students, after having understood the general MOOC structure, let us now try to know what will be the weekly layout for this course on forensic biology and serology by the MOOC coordinator Dr. Adarsh Kumar.
Dear students, welcome to this course on forensic biology and serology for PG students. This course, which you are supposed to complete in 15 weeks, has been developed in a very systematic and logical manner. So, the first week will give you an overview of forensic biology and serology, wherein you will be studying about the various types of biological evidences such as blood, urine, saliva and semen, their compositions and importance in the field of forensics. Moreover, you will also be made familiar with how you can locate stains from these biological evidences. In addition to it, you will also be studying about nature and role of forensic biologists in crime scene investigation, in which you will be studying about how to protect, recognize, search and secure the biological evidences in crime scene investigation. This will be followed by the second week, in which you will be studying about how to handle biological evidences. In this week, you will be studying in detail the forensically important biological evidences, for instance blood, semen, vaginal secretions, feces, etc. Similarly, the second chapter of this week will focus on how to collect and preserve biological evidences and also what are the precautions which are required to be taken during preservation of such evidences. The third chapter completely focuses on how evidence collection from the victim, suspect and crime scene is carried out, and also about clean collection practices. You will also get to know how biological evidences are packaged, documented and stored. In the third week, you will be studying about forensic examination of semen, in which you will get to know more about the composition of semen, structure of spermatozoa and also the various functions performed by its different parts. Furthermore, in this week you will also be studying about how examination of semen and seminal stains can be carried out. For instance, physical examination, the visual examination, the microscopic examination. Also, we will be telling you what are the confirmatory tests required in cases of seminal fluid, and also about the application of forensics in semen examination. The fourth week, entitled as Forensic Examination of Saliva and Urine, will emphasize on saliva, its composition, and the different type of tests, namely starch iodide test, Fadibas test, etc. Furthermore, you will also be studying about urine, its composition, the presence of different components such as urea, nitrate, creatinine, indican. In addition to this, you will also be studying about how screening for presence of drugs in urine can be done. Students, after you have studied about some of the biological evidences, for example, semen, saliva and urine in detail, the fifth week will emphasize on forensic examination of sweat and fecal matter. Sweating is a process in which fluids are secreted by the sweat glands in the skin of mammals, whereas excretion is a process in which human waste is thrown out of the body. Also, because they are important from the forensic point of view, that's why you will be studying about these in detail. Moreover, you will also be studying about how sweat stains and fecal matter can be examined for and how they are forensically important. The most common type of biological evidence which may be found at a crime scene is blood. So, keeping this in mind, the sixth week has been dedicated to teaching you regarding the forensic examination of blood. Blood is a complex fluid tissue composed of a liquid portion, plasma and cellular components. You will also get to know that how identification of origin of blood stains can be done by methods such as Octerluni double diffusion method, rapid immunoassay methods and many more. Moreover, you will also be studying about the preliminary and confirmatory examinations of blood, how age of blood stains is determined, and what are the factors which affect the detection of blood from blood stains. In the seventh week, you will get to know about hair examination. Although hair, being a trace evidence, is also considered a biological specimen, and hence in this week you will be studying about the examination and analysis of hair. Students, in this week you will be studying about structure of hair, how body area determination is done, differentiation between human and non-human hair, determination of race and origin, and also about the forensic importance of hair examination. The 8th week shall be a revision and assessment week for you. In this week, you are supposed to revise what all you have learned in this course over the past 7 weeks. We will give you assignments which you are required to submit in time. Please note that the marks obtained by you in these assignments shall count towards your grades in this course. The most vital principle in forensic analysis is the well-known Locard principle of exchange. It suggests that every contact leaves a trace. In forensic analysis, fiber acts as important evidence where contact between two or more surfaces is suspected. Thereby, with this view, the 9th week has been assigned towards making you familiar with the types of fiber and their comparison. So, in this week, you will be learning about what is fiber, its classification, comparison between synthetic and natural fiber, and also about the significance of fiber. Coming on to the 10th week, we will be taking up examination of fiber and dye analysis. In this week, you will be studying about how physical and chemical examination will be carried out. In addition to this, you will also get to know in detail about how analysis of dyes is carried out by thin layer chromatography, high performance liquid chromatography, and also the capillary electrophoresis. In the 11th week, you will be studying about genetics and DNA markers, wherein you will also get to know about cell, nucleus and chromosomes. In addition to it, you will also be studying about Mendelian genetics and how genetic variation takes place. Also in this week, you will study about structure of DNA, nucleotide composition and some of the salient features of DNA. This will be followed in the 12th week where you will learn about biochemical genetics, gene mapping and expression. In this week, you will be studying about inherited metabolic or genetic disorders, about gene mapping, types of map, DNA sequencing, the central dogma, transcription, translation and much more. In the 13th week, you will get to know about forensic significance of genetic markers and matrices supporting protein electrophoresis, wherein you will be studying about types of genetic markers, biochemical and molecular markers, about techniques such as RFLP, SNP, SSR. Also in this week, you will also be studying about matrices supporting protein electrophoresis and about various polyacrylamide gel electrophoresis techniques. The 14th week, which will be the last part of this course, will be about study of erythrocytes, protein, hemoglobin and serum, wherein you will be told about erythrocytes, blood proteins and also their forensic applications. Furthermore, you will also be taught about hemoglobin, how mutation occurs, about sickle cell anemia, thalassemia. In conjunction with this, you will also get to know about protein profiling, its techniques and advantages. The 15th week shall also be a revision and assessment week for you. Here also you will be given assignments which you are required to submit in time. After the completion of this course, there shall be a proctored examination about which you will be notified at appropriate time. So dear students, I am sure you will find this course very interesting and enjoyable. Please go through the entire content which includes self-learning videos, e-texts and learn more etc. very thoroughly and perform the various activities and assignments of this course sincerely. I am quite confident that after going through this course will be a great learning experience for you. I wish you all the best.